So we will start today extracellular matrix. Now we have seen there is tissue and in between the cell, what is there? There should be something, because in tissue cells are arranged in clumps, but there is a space in between the cells, that is the extracellular space, and that is not empty. There is something in that extracellular space that is called extracellular matrix. Extracellular space, which is a gel-like substance in the extracellular space. This is also known as connective tissue or ground substance. Connective tissue, also known as connective tissue, and so this is gel-like substance present in extracellular space, also known as ground substance or connective tissue. From that, can you see? Can you understand that? Okay. Now, components of extracellular matrix, we can divide into components. We can divide into three groups. Okay? Structural proteins, then specialized glycoproteins, and proteoglycans. Broadly we can say it has three components: structural protein, specialized glycoproteins, then proteoglycans. In structural proteins, the major component is collagen. Then elastin, it's not elastin, it is elastin. Elastin, then fibrillin. Specialized glycoproteins, these are fibronectin and laminin. This is collagen, elastin, fibrillin. In proteoglycans, they are agrican, perlican, syndican, sarglycin, decorin, beta-glycan, fibromodulin. Each of them has a specific function. So many these specialized products are there in the extracellular matrix. They are responsible for different functions of cell. Now, extracellular matrix we can broadly find two types. One is basement membrane, another is gel matrix. So here these are the cells, and this is the collagen network on which these cells rest. This is called basement membrane, and inside these things, these are the stromal matrix. They contain cells, proteoglycan, this light green one, proteoglycan gels. Then here and there you can find fibronectin, collagen fibrils. They are in the stromal matrix. So, two basic forms: interstitial matrix and basement membrane. Interstitial matrix, they are present between cells in connective tissue. It is synthesized by the mesenchymal cells like fibroblast. Major constituents are fibrillar and non-fibrillar collagens. Also contain fibrin, fibronectin, elastin, proteoglycan, hyaluronic and other elements. Now, basement membrane, it lies underneath the epithelium. It is synthesized by overlying epithelium and underlying mesenchymal cells. Major constituents are amorphous non-fibrillar type 4 collagen and laminin. Type 4 collagen, they form a mesh-like thing, and there is laminin, and on that this cell they rest. Now, this is an overview of extracellular matrix. This is the proteoglycans. These are blood vessels. These are collagens. This is the mesenchymal cells, macrophage, mesenchymal cells, that means fibroblasts, and they are responsible for synthesizing the matrix materials. Components of ECM we have told here. Functions of extracellular matrix broadly: it provides supports and anchorage of the cell, provides support, regulates and determines cell dynamic behavior, polarity of the cell, cell differentiation, adhesion, migration. Each cell is anchored with the extracellular matrix component. So, they have a fixed location, they cannot move from here and there. So, once there is differentiation of the cell, where they will stand, where they will go, it is already defined. So, it cannot just move here and there. Provides mechanical support for tissue and organ architecture. Mechanical support for tissue, say bone, it is a weight-bearing tissue. So, it needs a strong, it should have a capacity to bear the weight. So, that is provided by the component of extracellular matrix, that is one component is collagen. Okay. Now, it regulates growth because growth factors, they entrap in the extracellular matrix and release whenever required. Regenerative and healing process, once there is cut, that is being healed up by the fibrous tissue. Okay. So, that is provided by the extracellular matrix. Determination and maintenance of the structure, cells, tissue, organ structure, how it will be, what will be its shape, and this shape will be maintained by the extracellular matrix. Place for active exchange of different metabolites, ions and water. You know that things happen in the extracellular matrix also, because some ions, they are predominantly present in the extracellular matrix, some ions, they are present predominantly inside the cell. Okay. Now, we know that there is sodium content of extracellular matrix is very much high, because extracellular matrix components are negatively charged, so they attract sodium. So, where there is sodium, there is always water, is not it? So, a hydration is maintained by the extracellular matrix both for itself and to the cell. Okay. So, whenever there is decrease in potassium concentration inside the cell that is supplied through the extracellular matrix. So the intracellular potassium ion concentration is maintained. It is an example. Like that, many ions then metabolites, these are regulated and exchanged through extracellular matrix. Now, major component of extracellular matrix is collagen. Now we will go for synthesis of collagen and its function also. So collagen is a major component of extracellular matrix, constituting about 30% of the total weight of extracellular matrix component. This is a structural protein. Collagens are composed of 3 separate polypeptide chains. There are 3 polypeptide chains wound around each other like a rope. Okay. In collagen, it is triple helix wound around. About 30 collagen have been identified. Out of that, collagen 1, 2, 3 and collagen 4 are very important. They are denoted by Roman numericals. So around 30 types of collagen has been identified. Now, according to source, depending on the source, there are 4 types of, these 4 types of collagens are very important. Collagen type 1, it is present in bone, cornea, connective tissue, skin, tendons, cartilage. Collagen type 2 is present in cartilage, vitreous humor. Collagen 3 is elastic tissue of lung, blood vessel, skin, tendon. Collagen 4, glomerular basement membrane of in basal lamina. According to the structure, they can be divided into 7 groups: fibril-forming, network-like collagen, facet, fibril-associated collagen with interrupted triple helices, beaded-like, anchoring, transmembrane, and others. Just know the names. Fibril-forming, these are in tendons. They are arranged in longitudinally and like strands. No? So these are fibril-forming. Network-like collagen, it will create a mesh-like thing. Now, collagen, it is synthesized by the fibroblast. Muscle cells and epithelial cells also synthesize to some extent. It has 3 alpha chain. So it has 3 alpha chain. It is wound around each other. Collagen is having repeated units of glycine, X, Y. So this has 3 alpha chains. Each chain is composed of repeated units of glycine, X, Y. Y is proline or hydroxyproline and X is any other amino acid. Okay? So it will have again glycine, X, Y, like that. each alpha chain has around 1000 amino acids in its sequence. These 3 alpha chains, they are wound around and they are stabilized by hydrogen bond, disulfide bond and cross-linking. Now, this pre-pro-collagen, it is synthesized in the rough endoplasmic reticulum. Suppose this is a rough endoplasmic reticulum, here is the ribosome. So, in ribosome, this polypeptide alpha chain is synthesized. Now, this alpha chain in its amino terminal has a signal sequence, a specific amino acid sequence that is called signal sequence. So, that is after synthesis released in the rough endoplasmic reticulum. This is the signal sequence. So, rough endoplasmic reticular lumen. So, in the rough endoplasmic reticular lumen, the alpha chain containing signal peptide is termed as pre-pro alpha chain. In some book they have written pre-pro collagen. If you write pre-pro collagen, it is also okay. Now, if you write alpha chain also, it is also okay. But I think alpha chain, if we tell, it is more reasonable. Okay. Now, it is in the endoplasmic reticular lumen, rough endoplasmic reticular lumen. So, what happens with the help of signal peptidase, this signal amino acid sequence is cleaved off, it is removed. So, it is removed and the polypeptide chain without signal peptide is called now pro-collagen. Still it is in the lumen of the endoplasmic reticulum. So, in pro-collagen, there is hydroxylation. This is called post-translational modification. Post-translational modification. Hydroxylation. Disulfide bond formation. Glycosylation. Cross-linking. Now, out of this glycosylation and hydroxylation and disulfide bond formation that occurs inside the endoplasmic reticular lumen. Now, once these two are formed, once this hydroxylation and disulfide bond is formed. Then 3 alpha chain, they come together and form a triple helix. Okay. Now it is not that strong because some other things are supposed to occur. Now in hydroxylation, where does hydroxylation occurs? It is in the third amino acids, proline. In proline, there is hydroxylation by the enzyme prolyl hydroxylase. So here prolyl hydroxylase, again in third position there may be lysine, second position there may be lysine. So lysyl hydroxylase, these are the 2 amino acid responsible for hydroxylation, and they need ascorbate as cofactor. Ascorbate as cofactor. So what is the need of hydroxylation? Because to stabilize the triple helix, a lot of hydrogen bond is required. So hydrogen bond can form with the amino acid having hydroxyl group. So there is hydroxylation, extra hydroxylation of proline and lysine. We have 3 amino acids already in the chain having hydroxyl group: serine, threonine and tyrosine. So with these two we have total 5 amino acids. So in the chain there will be lot of hydroxyl containing amino acid giving strength through hydrogen bond so that the triple helix structure can be remain stabilized. Now so here this is the nucleus. Here mRNA is synthesized. mRNA is the template for alpha chain synthesis. So mRNA will come in the rough endoplasmic reticulum. In the wall of the rough endoplasmic reticulum there is ribosomes. In the ribosome these alpha chain is synthesized and released inside the lumen. So in the lumen there is hydroxylation and disulfide bond formation. So the pro-collagen, once there is hydroxylation and disulfide bond formation, the three alpha chain, they will come together and form triple helix. So here we call these are pro-collagen. Pro-collagen. Still it is in the endoplasm reticular, rough endoplasmic reticular, inside the rough and endoplasmic reticulum. So this pro-collagen triple helix, they pass to the Golgi. They enter through cis Golgi, cross through the middle Golgi, and they get glycosylation and cross-linking. They undergo glycosylation. So now the triple helix is glycosylated and there is formation of tropocollagen. Now after glycosylation it becomes tropocollagen. So it is now tropocollagen. Now here, now just see that both end of triple helix, they are a bit bulged due to the excessive cysteine containing amino acid. So when it is shifted to the extracellular matrix, there is action of two enzymes, the amino peptidase and carboxy propeptidase. So these two ends, they are clipped off, the bulge two end. Okay. Now it is protocollagen. Now protocollagen, they are in the extracellular matrix. So they need more tensile strength. So here two things happen. There is cross-linking of the collagen fibers. Now there are two reaction happens. It is by skip base formation or aldol condensation. Skip base formation is lysine residue. It undergoes deamination by lysyl oxidase. By lysyl oxidase and forms aldose. Then these aldose, it reacts with second lysine residue and forms skip base. Here is the skip base formation. And when these aldose reacts with another aldose, they form aldol concentration. By this two reaction, the collagen cross-linking occurs, and this gives the maximum strength of the collagen, and the topocollagen is now termed as collagen. This is the mature collagen. Here is the cleavage by the amino terminal, in the amino terminal by the amino peptidase, and in the carboxy terminal it is carboxy propeptidase. Now its function is collagen contributes the stability of the tissue and organ, maintains structural integrity, has great tensile strength, main component of fascia, cartilage, ligament, tendon, bone and skin, plays an important role in cell differentiation, polarity, movement, plays an important role in tissue and organ development. Now, various disorders associated with collagen. Number one is osteogenesis imperfecta. Osteogenesis related to bone development, imperfecta means imperfect bone development. So this is osteogenesis imperfecta due to the mutation of gene of type 1 collagen. Osteogenesis imperfecta defect in type 1 gene responsible for synthesis of type 1 collagen. Okay, it is characterized by, as it is a genetic disorder, gene mutation is there, genetic disorder. So there will be a family history. There is positive family history of similar type of defect or illness. Okay. Now it is characterized by brittle bone. It is the central feature of the osteogenesis imperfecta. Brittle bone. Very easily the bone is broken because there is collagen formation is defective. Collagen, it gives the strength. So once there is defective formation, then it will not gain strength. It is associated with the deafness, blue sclera. Blue sclera means your eye white is there. Eye white is there. So it is collagen. Eye white, that part is collagen. So once there is a less collagen formation, that white part becomes very thin. Inside that white sclera, there is choroids. So through sclera, choroid becomes visible. That's why the sclera appears blue. Okay. Now, like that of bone, our teeth also, it is made up of calcium and it has a great strength. So naturally, there will be dental defect. Dental abnormality will be there. Now, second is Ehlers Danlos syndrome. This is mutation because of the mutation of the gene responsible for synthesis of type 1, type 3 and type 5 collagen, and may be associated with lysyl hydroxylase malfunction. There is hypermobility of the joint. So we can extend up to this much. But in case of Ehlers-Danlos syndrome, they can extend beyond this. Okay. So there is hypermobility of the joint, hyper extensibility of the fingers. Fingers, they can extend like that. Okay. So there will be skin deformity, like valvulate skin will be there, hyper extensibility. To some extent we can lift the skin, when release it goes back. Okay, they can pull it more. Cigarette paper scar, when some part of the skin is injured, that is healed up by scar formation. Okay, as there is no collagen formation, scar is nothing but the aggregation of collagen. Okay, now once there is less collagen, then there will be less thin scar I should say. Okay, so there will be cigarette paper like healing of scar. Cigarette paper very thin, so the scar is also very thin. There is stress mark in skin. Stress mark usually commonly we find after pregnancy. Then those who are very thin, lean and thin in the childhood become very fatty in the when they grow up. So there is stress mark in the skin, in the thigh, then lower abdomen. Okay, those who become suddenly fatty, they get stress mark in the skin. So these also, they also will have stress mark because the skin elasticity is very less. The next is Alport syndrome. Alport syndrome, it is due to the defect in gene coding type 4 collagen. Type 4 collagen, they are abundantly present in the glomerular basement membrane in the lamina. So there will be renal failure and hematuria and renal failure. Okay. Now next is Menkes disease. Menkes disease, Menkes kinky hair disease. Okay, Menkes disease, defect in gene encoding ATP7A gene causing defective utilization of the copper. Copper, it gets absorbed from the intestine, but from the intestinal lumen epithelium, it is not absorbed in the blood. It is not shifted in the blood. So peripherally it is not available for utilization. Okay. So in the enzyme lysyl oxidase, what we told in the skip base formation, lysyl oxidase enzyme is there for skip base formation and aldose condensation. So there will not be cross linking. There is defect in cross linking because of the absence of the cofactor copper, responsible for normal activity of the oxidase, lysyl oxidase. Okay. So aldol concentration and skip base formation will be abnormal. They are characterized by kinky hair, thin, brittle hair, thinly sparse, brittle hair. Okay. Then skeletal deformity will be there. Then neurodegeneration, the lysyl oxidase, similarly dopamine in the metabolism of tyrosine, we have seen epinephrine formation, dopamine oxidase is there, that also require copper. So that also will be defective in its action. So there will be mental neurodegeneration. Okay. Now this is a picture of osteogenesis imperfecta. See the condition of bone. This is the femur of the baby. See, this is band, like multiple band, no? And the cortex of the bone is very thin, so it is easily fragile. See, it is broken here. Okay, it is easily gets broken. This is the Menkes kinky hair, sparsely arranged and fragile hairs. Next disease is scurvy. Vitamin C is required for the function of lysyl hydroxylase and polyl hydroxylase. So once there is deficiency of vitamin C, hydroxylation will not be properly taken place. So there will be less hydrogen bond and weak collagen formation. Okay. The defective hydroxylation is due to the defective polyl and lysyl hydroxylase. It is characterized by capillary fragility. Capillary fragility, easily bruise can occur. Bruise, you know, bruise means the bleeding underneath the skin. If you get trauma in any part of the body, it gets swollen, blue color. Okay, it is because of the collection of blood in the subcutaneous tissue. So in scurvy, easily they get bruised. Even if you hold like that, they may get bruised. Okay. So there may be hemorrhagic tendency, brushing the teeth, there is bleeding. Then gums, it is bleeding gums is there, gum is there. So due to the vitamin deficiency, abnormal collagen formation is there, and that causes bleeding, capillary fragility problems. Next comes epidermolysis bullosa. Epidermolysis bullosa. This is because of the mutation of collagen genes responsible for collagen 7 synthesis. There is easy rupture of skin and blister formation with minor trauma. Spelling mistake is there. Minor trauma. Next component is elastin. The ability of the tissue to recoil and return to the baseline structure after physical stress is conferred by the elastic tissue, especially important in the walls of the vessels like aorta, uterus, skin, ligament. elasticity and recoiling of the lung and large vessels like aorta and tendon. There is in elastin, there is no triple helical structure. Note that there is no triple helical structure. There is no formation. It is formed from the proelastin. There is only hydroxylation of proline, but not lysine. Only proline is hydroxylated in elastin. This is a simple polypeptide chain it is having. Now disorders are Williams syndrome. Williams syndrome, this is due to gene coding the elastin. There is supravalvular aortic stenosis. Supravalvular aortic stenosis. Okay. Now in heart there are valves. Have you heard of heart? Did you so this is the left. Left ventricle, from left ventricle aorta arises. Okay. Now aorta, this is the aorta. So there is a valve here. That will prevent blood from coming back to the heart. Heart will push the blood, it will go to the aorta but cannot return back to the heart. So just above the valve there is a dilatation, stenosis. There is a stenosis due to the less elastin formation. So there is the space for ejection of blood becomes narrow. Okay. So there is supravalvular aortic stenosis. Now there is scleroderma because of the deficiency of elastin. In scleroderma there is increase in elastin level. So once there is increase in elastin level, there is a increased tensile strength. No. So the patient of scleroderma, the patient finds difficulty in opening this smiling. Okay, he will do like that. No, he cannot smile freely. Okay. Now due to the increase this one elastin, the skin it cannot be pulled freely. So there is stiffness of the skin. Then emphysema, there is increased destruction of elastin. So recoiling capacity of the lung is very much reduced. So patient lung becomes swollen. It gets enlarged. No? Enlarging the lung means capacity to inhale air becomes less. So there is always respiratory distress because required amount of oxygen cannot be taken inside. Old age due to the problem of aging, there is less formation of elastin. Now fibrillin, it is present in periosteum, aorta, eyes. Defective gene for fibrillin 1 leads to the Marfan syndrome. Marfan syndrome is characterized by long digit. Fingers are very long. Yeah? Long digit, tall stature, very tall. Then ectopia lentis, defective lens, dilatation of the aorta. Fibrillin is present in the aorta also. So there will be dilatation of the aorta. Now coming to the second group, specialized glycoproteins. They are fibronectin, laminin. Fibronectin, its function is cell adhesion, cell migration. It is a large glycoprotein. Decrease in fibronectin causes metastasis because cell adhesion with the extracellular matrix component, it prevents moving the cell from here and there, migrating the cell, unprogrammed migrating the cell from here and there. In case of malignant tumor, this fibronectin becomes defective and cannot hold the tissue in its place anymore. So tissue means malignant tissue, malignant cells. It can move easily, go to the distant place and metastasize, produce tumor there also. So this is called metastasis. Laminin present in the laminar basement membrane. Thick membrane responsible for glomerular filtration, contains heparan sulfate in plenty, so it is a negatively charged membrane. If there is infection in the glomerulus, there is loss of heparan sulfate, so a negativity of the basement membrane is lost and the molecule can pass through it. One of the molecule is low molecular weight albumin. So in case of glomerulonephritis, there is albuminuria. So also in nephrotic syndrome, like proteoglycans, the third group, proteoglycans. So proteoglycans made up of core protein attached it with, there is a core protein, hyaluronic acid. See, there is a hyaluronic acid attached with a core protein, long one. Now within that core protein, the glycosaminoglycans are attached. Okay. Now this is the structure of proteoglycans. The proteoglycans which are abundantly present in the glycosaminoglycans which are abundantly present in proteoglycans are hyaluronic acid, keratan sulfate in the corneal tissue, chondroitin sulfate in cartilage, dermatan sulfate in skin, heparan sulfate in glomerular basement membrane, and heparin which act as anticoagulant. Okay. Now disorders of proteoglycans, they are collectively called mucopolysaccharidosis. So individual diseases are Hurler syndrome, Hunter syndrome, Sanfilippo A, B, C, 4 types are there. Morquio syndrome A and B, Morotox disease, Slough. Very rare disease these things, but we have to know. General feature is there will be skeletal deformity, organomegaly, there will be mental retardation and visual impairment and deafness. So we have read a 1 hour, very big thing, so many things. No? But you will get question. The question you will get a short answer type of question, extracellular matrix. What you will get? You will get definition, types of extracellular matrix. If it is short note extracellular matrix, write the definition, types, its function and the main components. Okay, you don't have to go all these things. Now you will get short note as collagen. So if you get collagen, then say what is extracellular matrix? Abundantly present material is collagen. Then mention 2-3 function of collagen and very shortly the collagen synthesis. Pre-pro-alpha chain, pro-collagen, okay, then tropocollagen, mature collagen. Okay. Now just mention what are the things which hampers collagen. Disease you may not mention in short note. Okay. So these are the things. So thank you.